Okay, this is going to be the first part of the test review, and I've got my review. And just to make it quick and easy, I know there's another video showing you how to do it all fancy the way I've got it in my notebook, but when we're actually filling it in, I've just folded it. I folded it in half like that, and then I folded the middles in like that, and all I'm going to focus on are these four here to start off with. So excuse me, this makes up about half of your tests, because this is the big things: monohybrid crosses, dihybrid crosses, and meiosis. So let's start with meiosis. So here is a somatic cell. How do I know it's a somatic cell? The chromosomes are in pairs, and the word we use for chromosomes in pairs is diploid. Sometimes that's written as 2n. So in humans, the diploid number of chromosomes, 2n, is 46. And you'll notice we go through a cell division and then a second cell division. And what we end up with is 1, 2, 3, 4. That is a key that you're looking at meiosis and not mitosis. You start with one cell and you end up with 4 cells. And these cells are gametes. G-A-M-E-T-E-S, gametes, which we know are sex cells. Okay, so drew some little spermies there, and maybe an egg cell. Okay, gametes. And the number of chromosomes in gametes is half of what it is in the original cell that produced them. And the word we use for the cells being halved is haploid, and sometimes that is written as N. So in humans, N is 23, because there are 23 chromosomes in each gamete, whereas the diploid number of the body cells, like muscle cells and skin cells and nerve cells and stuff like that, in humans it's 46. The diploid number, 2n, is 46. So 2n is actually double the number of chromosomes you find in a gamete. Okay, so one cell becomes 4 cells. Big clue that what you're looking at is meiosis. Okay, another key thing in meiosis is this: during prophase 1, homologous chromosomes. So remember when we did the pairs of chromosomes, we cut them out, we organized them, we did that karyotyping exercise. So you have a chromosome from mum and a chromosome from dad. That is what this would be. These are homologous pairs. And during prophase 1, they kind of crisscross with each other like this, okay? And when they uncrisscross, what you see is they have traded out parts with each other. So now the chromosomes do not look, they started off all green, all black. Now they kind of they're sharing bits of each other. That is referred to as crossing over, okay? And it is a very important element of meiosis. In fact, the actual objective is to recognize the importance or the significance of meiosis to sexual reproduction. And there are 3 elements of that. Number 1, we produce gametes, right? You could not have sexual reproduction if you did not have sperm cells or egg cells, or if you're a plant, pollen and egg cells. Okay, so it produces gametes. That's number 1 reason it's important. Number 2 reason, we have the number of chromosomes. Remember, what does a sperm cell want to do? A sperm cell wants to meet an egg cell. So if we don't decrease the number of gametes by meiosis, and the sperm cells had the same number as any other body cell, at fertilization, that number would go from 46, it would go to 92, it would double. We don't want to do that. We want a fertilized egg cell to have 46 chromosomes in it. So if there's 23 from the sperm cell, 23 from the egg cell, we get the right number. We get 46. So it reduces chromosomes by half. So we go from diploid to haploid. That's one of the three elements. So we produce gametes, we have the number of chromosomes, and the other thing is crossing over produces genetic variation, genetic diversity. That is why you look similar but not identical to your siblings. So meiosis, start with one, end up with 4 gametes, crossing over during prophase 1. We produce gametes, we reduce the chromosome number, and we produce genetic variation. Okay, that is the big high points, the key points of meiosis. Now, let's look at this one. This is a monohybrid cross. Okay, monohybrid because, well, two reasons. One, the main reason is we're only dealing with one single characteristic, and the other reason it's a monohybrid cross is because we have a Punnett square that's got 4 boxes in. And this is what you're going to have to do. And when you have a cross in a test question, if you do not jot down a Punnett square and fill it in, you will mess it up. It is the easiest thing in the world to mess up a genetic cross by taking a shortcut that you don't need to take. You always need to draw the Punnett squares. You always need to work out what the gametes are, what the parent genotype is. This one says solid color fur, big S, is dominant to ticked fur, small s, in cats. It does not matter if you do not know what ticked fur is. It is irrelevant. Every genetic cross is going to have stuff that you've probably never heard of before, and that's fine. The principle, how you do the cross, is the same. So this one says, what percentage would have ticked fur? I know that if ticked fur is the recessive allele, the only way I can have ticked fur is if I am homozygous recessive, little s, little s. And so this one's easy because the Punnett square is already done. So big S, that one's going to have solid fur. Big S, little s, that's going to have solid fur. Big S, little s, that's going to have solid fur. And this one, little s, is going to have the ticked fur. So that is the only one where I'm going to have ticked fur. 1 out of 4, which equals 25%. Okay? And that's what you do for every monohybrid cross. You may have to draw the box out. You may have to do the actual cross. It may be already done for you, but you have got to fill the box in, just like we've been doing in class. All right, so here is an example of a dihybrid cross. How do I know it's a dihybrid cross? Because I'm talking about two different characteristics. And in this case, looks like I got little bugs here. It says normal wings is big N, little n is crinkled wings. You see it all crunched up. Red eyes is big R, that means that must be dominant. Little white eyes is little r, okay? I have red eyes. Okay. And then I've got a cross here. Big N, big R, little r. It's crossed with little n, big R, big R. Which trait do all the offspring have? So, remember how we talked about doing all the combinations? So I've got to have one of each letter. So I could have a big N and a big R, couldn't I? Okay? But I could also have a big N and a little r. And I could have a little n and a big R. Or finally, a little n, and a little r, like that. Okay? When I come over here, what are my options? I've got a little n and a big R. Little n, big R. And there's nothing else. There are no other options. So I don't need to use any more of these boxes. So looking in here, I can have big N, little n, big R, big R. Well, big N, little n is going to have normal wings, norm, and it's going to have red eyes. This one here, I got a big N and a little n, and a big R and a little r. Big N, little n, that's going to be normal, isn't it? Big R, little r, that's going to have red. So I still don't know which one's they're all going to have. Now looking here, I've got a little n and a little n. I've got a big R and a big R. Well, little n, that comes back up to here. So that's going to have crinkled wings and big R, big R. It's going to have red eyes. I think I probably know what my answer is now. If I look at the bottom one here, that's a little n, that's a little n. Little n, little n. That's a big R and a little r. So big N, little n is crinkled, right? And big R, little r would be red eyes. So by working out my options, one of each letter, doing the Punnett square, I now know that 50% of them will have normal wings and 50% will have crinkled wings, but 100% of them will have red eyes, which means, which trait do all offspring have? They all have red eyes. Okay? I didn't take a shortcut, and I'm guaranteed I got the answer right. Okay, that makes up about half of the test, that skill set. So I'm going to stop my review here, and we will pick up the next section in the next video.